Welcome to this lecture on the fundamental world of nucleic acids and their structure. If you have ever wondered how a tiny, microscopic cell knows exactly how to build a human being, or how traits are passed from parents to children, you are in the right place. This topic is the absolute foundation of genetics, molecular biology, and modern medicine. By the end of this session, you will understand exactly how life stores its most critical secrets. To start our journey, imagine you are trying to build a massive, incredibly complex Lego spaceship. To do this successfully, you need a highly detailed instruction manual that tells you exactly which piece goes where and in what order. In the human body, and indeed in all living things, we have a similar instruction manual that tells our cells how to build everything we need to survive. This biological instruction manual is made of molecules called nucleic acids, which you probably know as DNA and RNA. Just like a book is made of sentences, words, and letters, nucleic acids are made of long chains of smaller building blocks. Now that we understand the purpose of nucleic acids, let us look at their formal chemical definition. Nucleic acids are large, complex biomacromolecules. They are polymers, meaning they are long chains made up of repeating smaller units, called nucleotides. You can think of nucleotides as the individual links in a long chain. Each individual nucleotide is composed of 3 distinct parts: a 5-carbon sugar, which acts as the core, a negatively charged phosphate group, and a nitrogenous base, which acts as the actual letter of the genetic code. Interestingly, when just the sugar and the base join together without the phosphate, it is called a nucleoside. But once that phosphate attaches, it becomes a complete nucleotide, ready to build a nucleic acid. Building on the structure of a nucleotide, let us zoom in on the nitrogenous bases, as they are the most critical part for storing information. These bases are divided into 2 main categories based on their chemical structure: purines and pyrimidines. The purines include adenine and guanine, and they have a larger double ring structure. On the other hand, the pyrimidines include cytosine, thymine, and uracil, which have a smaller single ring structure. The specific sequence of these bases along the nucleic acid chain is exactly what spells out the genetic instructions, much like the sequence of letters on a page spells out a story. Knowing these chemical structures might seem abstract, but this knowledge has profoundly shaped our modern world. The structure of nucleic acids is arguably the most important architectural blueprint in all of biology. Without DNA and RNA, cells would have no memory of how to function, how to reproduce, or how to build the proteins that do all the physical work in our bodies. In the real world, understanding this structure has revolutionized medicine, agriculture, and forensic science. For example, when scientists design mRNA vaccines, like those used for COVID-19, they are directly using their knowledge of nucleic acid structure to teach our bodies how to fight a virus. Furthermore, DNA fingerprinting in criminal investigations relies entirely on the unique sequence of nucleotides present in a suspect's nucleic acids. This brings us to a fascinating mathematical property of DNA discovered by Erwin Chargaff. When we look at the double-stranded structure of DNA, the nitrogenous bases on one strand pair specifically with the bases on the opposite strand. This pairing is not random. Adenine always pairs with thymine, and guanine always pairs with cytosine. Because of this strict pairing mechanism, the total number of purines must exactly equal the total number of pyrimidines. This is mathematically expressed by Chargaff's rules. The formula simply states that the percentage of adenine is always perfectly equal to the percentage of thymine, and the percentage of guanine is always perfectly equal to the percentage of cytosine. Because these four bases make up the entirety of the DNA sequence, their combined percentages must always add up to exactly 100%. If the amount of adenine increases, thymine must increase by the exact same amount, forcing guanine and cytosine to decrease. Let us see this formula in action with a concrete example. Imagine a scientist isolates double-stranded DNA from a newly discovered bacteria and finds that 22% of the bases are adenine. We need to calculate the exact percentages of thymine, guanine, and cytosine. First, we use the rule that adenine pairs with thymine. Since adenine is 22%, thymine must also be exactly 22%. Together, they make up 44% of the total DNA. Next, we subtract this 44% from the total 100%, which leaves us with 56%. This remainder belongs entirely to guanine and cytosine. Finally, since guanine and cytosine must be present in equal amounts, we simply divide 56% by 2. This gives us 28% for guanine and 28% for cytosine. We have successfully mapped the entire base composition of this bacterial DNA. Now that we have calculated the base composition, let us understand what those numbers physically mean for the bacteria. In our previous example, the DNA had more guanine and cytosine pairs than adenine and thymine pairs. In the real world, this often means the bacteria's DNA is more stable at higher temperatures. This stability occurs because of the chemical connections between the strains. Guanine and cytosine pairs share 3 hydrogen bonds, whereas adenine and thymine pairs only share 2. Therefore, a DNA molecule with a higher percentage of G and C bonds is physically stronger and requires more heat energy to break apart. This is a beautiful example of how mathematical composition directly dictates biological function. Moving beyond the percentages of bases, we can also calculate the actual physical dimensions of a DNA molecule. This is crucial for biologists to understand how tightly DNA must be coiled to fit inside a microscopic cell or viral capsule. The formula to find the total length of a DNA molecule in nanometers is L equals N times D. Here, L represents the total length in nanometers. N is the total number of base pairs in the sequence, and D is the physical distance between each adjacent base pair in the double helix. If the number of base pairs increases, the total physical length of the molecule increases proportionally. By knowing just how many base pairs are in a genome, we can determine exactly how long the uncoiled DNA strand would be. Let us apply this length formula to a real scenario. Suppose a small viral genome consists of a double-stranded DNA molecule that is exactly 5,000 base pairs long. We are given that the standard distance between each adjacent base pair is 0.34 nanometers. We need to find the total physical length in micrometers. First, we multiply the total number of base pairs, 5,000, by the distance per base pair, 0.34. This calculation gives us a total length of 1,700 nanometers. Next, we need to convert this result into micrometers. Since there are 1,000 nanometers in 1 micrometer, we divide 1,700 by 1,000. This shifts the decimal point 3 places to the left, giving us a final length of 1.7 micrometers. This tells us exactly how much space the virus needs to package its genetic material. To truly cement everything we have calculated, let us look at a visual representation of the DNA double helix. On the screen, you can see the classic twisted ladder structure. The continuous blue ribbons on the outside represent the sugar phosphate backbone, which provides structural support. The colorful rungs connecting the two sides are the nitrogenous bases we discussed earlier. Notice how the red adenine always pairs with the yellow thymine, and the green guanine always pairs with the blue cytosine. If you look closely at the connections between them, you will see dashed lines representing the hydrogen bonds. There are 2 bonds between A and T and 3 bonds between G and C, which explains the stability differences we calculated. Finally, the bracket on the right shows the 0.34 nanometer distance between each step of the ladder, which we just used to calculate the total length. We have covered a lot of ground today, so let us summarize the most important ideas. First, nucleic acids are the fundamental molecules of life, acting as the ultimate instruction manual for storing and transmitting genetic information. Second, we learned that they are built from repeating nucleotide units, each containing a sugar, a phosphate, and a nitrogenous base. Third, we explored how Chargaff's rules dictate strict base pairing, where adenine pairs with thymine and guanine pairs with cytosine. This strict pairing not only forms a stable double helix, but also allows us to mathematically calculate both the base composition and the physical length of the DNA molecule. You now have a solid understanding of nucleic acid structure, which is the crucial first step in mastering molecular biology. Great job!